Hi, everyone. Welcome to the next lecture on our unit on the killers. This week, we are going to be talking about the second leading cause of death in the U.S. So let's get into it. So the second leading cause of death in the U.S., not in a pandemic year, is cancer. And below heart disease, about almost 600,000 people die of cancer in a year. So in this lecture, we're really going to talk about the biology of cancer. What do we know about the biology of cancer? So first, there are 6 things that I really want you to remember about cancer. Cancer is the uncontrolled cell division of a single cell. So I think a lot of people think of cancer as this big, ugly thing that comes and we can solve cancer as one big thing, but it's really the uncontrolled cell division of one single cell in one person. So it is really hard to manage if it's coming from one single cell. It's really tricky disease, cancer. 2, cancerous cells have accumulated genetic damage through mutations. So we talked a little bit about genetics and things a little bit, but mutations in our genome, in our DNA. So cancerous cells have accumulated damage to their DNA is really what this says. Has accumulated those damages to DNA. And then 3 and 4, proto-oncogenes. So what does this mean? Proto-oncogenes are normal genes, so genes in our DNA that make normal proteins that promote cell growth. So proto-oncogenes are normally in our cells and they promote cell growth. And we normally want our cells to grow. You know, if we get a cut and we want our cells to regrow where that cut was. And proto-oncogenes do that. If we get a cut and our cells don't grow back, that would be bad. So proto-oncogenes are part of that. Tumor suppressor genes, on the other hand, are normal genes that we normally have that make normal proteins that inhibit cell growth. So why would we want to inhibit cell growth normally? Well, sometimes our cells are, they grow and grow and grow, and we're like, that's enough. We don't need as much growth. So if we got a cut, right, and our cells were filling up that cut, at some point we need to tell the cells that's enough. We don't need to fill that cut anymore. So that's where a tumor suppressor gene would come in. So a proto-oncogene would say, okay, go ahead, fill that cut. A tumor suppressor gene would say, that's enough, we're done filling the cut. That's how our normal cells would work. Number 5, at least 2 mutation events, and usually more, are required for cells to become cancerous. So I think a lot of misconceptions are that, like, oh, you can get a mutation and boom, you become cancerous, kind of like that X-Men or something in the movies where like all of a sudden you fall into this vat of, of juice of like green goo, and then boom, you become like a superhero, or you become like this cancerous blob. But it takes a lot, at least 2 mutations, sometimes more, to become cancerous or to become an X-Men. And 6, mutations can be hereditary, 10%, or caused by environmental factors, which is 90% of our mutations, or causing cancer. So, biology of cancer. So, let's define a few terms. What is a tumor? A tumor is a population of cells growing at abnormal rates. So, a tumor is just some cells that grow abnormally. It can be benign. So, an abnormal mass or abnormal cells remain at the site of the original tumor, or it can be malignant, where the abnormal cells start spreading to the other locations of the body. So, what makes this cancer? How does a tumor become cancerous? Cancer is really the growth of a malignant tumor cells, and failure of the mechanisms to control that cell growth. So if we look at this picture, we see our genetically altered skin cell. So remember, cancer is the disease of one cell. So this one cell got genetically altered, and it starts dividing rapidly and growing. And now it starts growing abnormally and starts to form a tumor or a population of abnormal cells. And those cells start to change form and start to grow that tumor. And that tumor stays in place. So now it's benign, or remains in the same place. So this is a benign tumor. It's in the same place where those cells grew. A malignant tumor or cancer is now it's breaking free and it's invading other places. Most likely it's starting to go where the blood flows, and can metastasize. So the cells are breaking off and flowing into the blood, and the blood takes it to other places in the body and creates a cancer. And those cells can then start growing uncontrollably other places. So that's the difference between a benign tumor, where it just stays where it was, and a malignant or cancerous tumor, where it invades other places and metastasizes. So, a little deeper into the biology of cancer. Let's talk about those 2 different genes that I had discussed, a proto-oncogene and a tumor suppressor gene. We're going to talk about them as a red light and a green light, or cell signals. So a proto-oncogene is really a green light. It's a signal that says, go ahead and grow. It's a green light that turns your cells on and says, this is a time, cells, that we would really like you to grow. It's a proto-oncogene. A red light is a tumor suppressor gene that says, now is not the time to grow. Stop. And what does that look like? So what if the traffic signals aren't working correctly? So what if those genes are the ones that are damaged by either heredity or environmental factors? So normally proto-oncogenes control normal cell division. It says, okay, it's time to grow. Go ahead and divide. This gene can get a mutation, either by environmental factors, some environmental factors we'll talk about later, but as an example, we'll use smoking. Smoking can be one of the major causes of cancer. So let's say you're smoking or somebody is smoking and it damages your DNA enough in one cell to mutate this proto-oncogene. And it now becomes an oncogene, which turns it on permanently. And it says, go ahead and divide. Just keep dividing the cell. So that it is now just completely a green light. So the cell is turned on for division. Well, then you would get cancer. Well, remember in my first slide, I said, well, you need 2 mutations to get cancer. That's because we actually have tumor suppressor genes. The tumor suppressor gene would come in and say, nope, you know what? We're going to stop here. It's okay. Red light. That's a no-go tumor suppressor right there. So we have backup. And so if you only have one mutation from smoking, you have a backup. You have a tumor suppressor gene that backs up and turns that off and says, okay, cancer, stop. We're going to stop that cell division. It's in that second mutation. Maybe maybe there's a lot more smoking going on, and the cell gets this tumor suppressor gene. Here's your mutation number 2. The tumor suppressor gene then no longer works. So then you get cancer. So your green light is now turned on and your red light no longer works. And that's where the cancer can grow uncontrollably. So our green light in our cells is turned on and it tells the cell, go ahead and grow. And our backup red light no longer works because it got mutated. And that's where the cancer can grow uncontrollably. So the biology of cancer, it really takes these 6 things. Cancer is an uncontrolled cell division of a single cell. Cancers have accumulated genetic damage through mutations, right? And I showed you 2 mutations. And really, a lot of the mutations are in these 2 genes, proto-oncogenes, which are responsible for normal cell growth, and tumor suppressor genes, which are responsible for inhibiting cell growth. And 5, you need at least 2 mutations, and usually more, are required for a cell to become cancerous. And mutations can be hereditary, so you can inherit those mutations from family, but a lot of mutations can be environmental. And we will learn about that soon.